Hello, everyone. I wanted to thank everyone for joining our Ignite Q&A webinar. Just right off the top, I would like to mention that this webinar is going to be recorded with the hope that we will be able to distribute it to everyone who is registered for this webinar and post it on our NINDS IGNITE website. The way that this webinar will work is that we're going to provide a brief presentation on the IGNITE program and the funding opportunities associated with the program. And then we're going to take your questions afterwards. The way that you're going to submit your question is in the Q&A field. You can find the Q&A box on the lower right-hand side of the WebEx. So just type in your question and then we will respond. And with that being said, I'd like to pass over to Dr. Roof to start the webinar.
Great, thank you, Tim. And thank you everybody for joining us for this IGNITE webinar. IGNITE stands for Innovation Grants to Nurture Initial Translational Efforts. It's an early stage translational program. I'm Becky Roof, I manage this program. My contact information is here. I also have with me Dr. Chardell Spriggs, who is a project manager for the IGNITE program, Dr. Julia Bachman, who's a health program specialist, Tim Lydon, who you just heard from, is our WebEx guru, and also Ashley Gibbons is our operations coordinator for the program. So the IGNITE program has the goal to get you ready for the next stage for later stage programs such as Blueprint or Create. It's meant to be a feeder program for these programs. Create is for later stage biologic development, and the Blueprint Neurotherapeutics program is for small molecules. Now, it's a really good idea to take a look at these later stage programs when you're writing your IGNITE grant, and make sure that you're writing your grant such that you're going to get to the entry criteria for these later stage programs if you're successful, okay? Because that's really the goal, is to get you to that point. All right, so in IGNITE, we have a suite of early translational opportunities. We have 3 FOAs, funding opportunity announcements. The first is PAR 18761, which is for neurotherapeutic agent characterization and in vivo efficacy studies. The second is 762, which is for assay development and therapeutic agent identification. And thirdly is 763 for the development and validation of model systems and or pharmacodynamic markers to facilitate neurotherapeutic discovery. We're going to talk about each of these in more detail later on, but up front, I'm going to tell you all 3 of them are 3-year projects. They have a budget limit of $750 direct costs for the entire project and a limit of $499 in any given year. We have a couple upcoming receipt dates, October 20th and February 17th. Now, I always get asked about late applications, so these are the relevant notices. The first one applies for situations like a serious illness or death in the family, but it also applies for those of you who are reviewers and qualify for continuous submission. We don't use continuous submission for these mechanisms, but you can apply 2 weeks late under this notice, but take a good look and make sure that it applies before you assume anything. The second notice, OD 20082, applies to people who need to apply late for COVID. So if you've been impacted by COVID, and I imagine many of you have been, this is something that you can consider. But again, take a really good look at them before you apply. And I'll also say that in both cases, we cannot promise or guarantee anything in advance. It's really a better idea to get things in on time if you can. So that way, at least for timeline reasons, you're not going to have any problems. Okay, now, all 3 of these FOAs use this R61, R33 mechanism. And this is new for a lot of people, so I'm going to take a minute to go over it. This is a phased mechanism. And the R61, you're going to get ready for the rest of the project. You're going to demonstrate feasibility, you're going to prepare, you're going to get your ducks in a row. And then there's a go, no-go milestone. And then if you're successful, and you meet your milestones, you move forward to the R33, which we're calling here the main event. But it might be in the first phase, you validate your assays so that in the second phase, you can do your high throughput screen. Or maybe in the first phase, you're doing your PK work so that you can interpret the proof of concept efficacy studies that you're going to do in the later phase, for example. Okay, so you can see how you're getting ready, you're demonstrating feasibility, and then if you're successful, you're moving on. Now you'll notice that this phased program allows for dependent aims. In fact, it's kind of designed to require dependent aims. It also allows for a little bit more risk, right? You can propose to do a high throughput screen in the second phase when you don't have your assays fully validated and ready to go because that's a first phase activity. Okay, the reason that you can do these things in this mechanism where maybe you can't do them in an R01 is because of this milestone, right? NIH and review are perfectly fine with these dependent aims and this risk because we know that it's only going to go forward if it's the first phase is successful, okay? These milestones are the keystone to this whole thing working, all right? So it's extremely important that they be really clear, that they be quantitative, and that they be definitive, okay? There shouldn't be any ambiguity around, have these milestones been met or not when we see the data, okay? They need to be able to support a go, no-go decision, and they need to reflect success of the first phase and that the second phase is worth doing, okay? So does this still warrant further effort now that we've seen the first phase data, okay? So that's really important, and it's a very different setup than what you might be used to if you've applied to R01s or R21s in the past. Now there's a single go, no-go decision point. Some other types of mechanisms will use multiple milestones. We have just one point. And the transition from the first phase to the second phase is done via an administrative review. And what that means is it's decided internally by NIH program staff. It doesn't go back out to peer review. We don't have to wait and set up panels and wait on that process. If you get everything in on time and there are no issues or problems, we can award your R33 without any lapse of funding. Okay, so it's a much faster, but because it's done internally and it doesn't go to peer review again, that's all the more reason that it needs to be clear and quantitative upfront. Now I'm going to harp on milestones for just a minute longer because this is something that is new to a lot of people and a lot of people have a hard time with. It's actually quite rare for somebody to not get a comment about their milestones in their summary statement. So I'm going to help you hopefully not have this problem when you submit your applications. I'm going to walk through some examples of weak milestones and strong milestones just to illustrate my point, what clear quantitative means. But I need to say we don't prescribe milestones in this program. You need to propose what makes sense for your particular project. These are just examples to illustrate my point. All right, so a weak milestone might be we're going to scale up compound A. A stronger way of saying that is we need to generate X grams of this compound. It needs to have this purity. And that's how we know we have enough that this is feasible to go forward. All right, a weak milestone might be we'll perform PK studies on several compounds and pick the best one. Is that really meaningful? If none of them look good, are you still going to pick the best one and go forward? No, a better milestone would be compounds must exhibit the following properties. This is how we know it's feasible to go forward. All right, a weak milestone might be we'll have fully optimized and validated assays. A stronger way of saying that is this is the z-score we need. This is the signal to noise ratio we need. Whatever other metrics are important for you in your assay, this is what we need. We need X, Y, Z. And that's how we know it's feasible to go forward. All right, treatment of protein B decreases seizures compared to control. Again, that's not quantitative. If you put that in your application and review said, no, that's not quantitative, define that. And then you came back and you could say, well, treatment of protein B decreases seizure control. That's statistically significant compared to control. Okay, now that's quantitative, but is it meaningful? Does it reflect success? If you saw a 2% decrease, but it was statistically significant, would that really mean anything? So a stronger way of saying that is no, we need to see a decrease by X%. for this long, okay? And by the way, somewhere in your application, you should talk about why is X percent meaningful. If a patient had a decrease in seizures by that percent, would they consider that meaningful? And you would also talk about with your N here, where did that come from? We have a power analysis, and we know that Z animals is going to give us the results we need. If we don't see it with Z animals, then we're done, okay? So it's clear, it's quantitative, and it reflects success. It reflects that this is worth going forward. All right, hopefully you can see the difference here. All right, we have more examples on our website. I believe you'll be getting these slides, and these links will be clickable. You can click on this, you can go to our website, you can see more examples. Also, when you contact us in advance to talk about your application and you send us an AIMS page, if your AIMS page includes milestones, that's perfectly all right, and we can talk about that when we set up a call.
Thank you, Becky. Now we're going to turn to Chardell Spriggs, who is going to talk about funding opportunity PAR 18761.
This FOA provides funding for studies to demonstrate that early-stage neurotherapeutics have sufficient biological activity to warrant further development. We use a combination of parameters, including target engagement, PD, PK, and in vivo efficacy studies to determine the feasibility of the candidate therapeutic agents. Entry criteria that are necessary and that you should be sure to illustrate in your application are outlined on this slide. The FOA seeks to apply new knowledge around targets, mechanisms, pathways, or therapeutic indications. Projects should aim to develop or repurpose therapeutics that are significant improvements over existing therapies to treat neurological disorders. We like to see a strong biological rationale for the intended approach, along with preliminary data or literature-based evidence supporting the feasibility and importance of the approach, including evidence that the therapeutic agent has the potential to be therapeutically viable, as well as evidence to support the robustness of the pharmacodynamic measures and/or in vivo efficacy models. All preliminary data should be from well-designed experiments. Projects should include a description of the unmet need for the therapeutic agent and a clear justification for how the findings from these studies are relevant to treatments for disorders that fall within the NINDS mission. Projects should explain how the candidate therapeutics will be optimized and advanced toward later stages of discovery once the R33 phase is complete. Next slide, please. Some examples of activities for the R61 phase, or the first phase of your award, include those listed below. While this is not an exhaustive list, it does give you a good idea of where to place your studies within the application. These include preparation of the therapeutic agents to support your proposed activities, characterization of therapeutic agents. This could, for example, include confirmation of purity, stability, in vitro absorption, distribution, metabolism, and excretion, in vitro potency and selectivity, degradation products, and process impurities. Any focused SAR to optimize the candidate therapeutic agent for in vivo efficacy testing, studies to develop dosage forms to support your proposed PD measures and/or in vivo studies, pharmacokinetic studies to determine therapeutic agent levels, any studies to confirm that therapeutic agents reach and engage the target site, whether directly or indirectly, and at a level that exceeds in vitro pharmacological potency over the desired period. Any studies to inform design and refinement of the PD measure and/or in vivo efficacy models and testing procedures, including examination of model variability to estimate sample size, calibration of the model according to positive and negative controls, along with the age, time course, and window of disease that are relevant for therapeutic testing. Next slide, please. At the end of the R61 phase, there are criteria that should be met to transition to the R33 phase. And this will occur if we have good milestones and your milestones are met. If this is the case, we should see successful completion of all necessary preparation and characterization of proposed therapeutic agent. This includes chemical synthesis and scale up to provide material for your proposed project, the in vitro and/or in vivo potency studies, in vitro organ tissue or cell culture systems, all pharmacokinetic studies, design, refinement, and validation with pharmacodynamic markers, using the appropriate positive and negative controls, and demonstrating feasibility of conducting therapeutic agent testing. And a further refined and detailed in vivo study design, such as in an animal model, that meets the NINDS rigor guidelines and will allow demonstration of dose and exposure response. Examples of activities for the R33 phase include, but are not limited to, any pharmacodynamic and/or in vivo efficacy studies with chemically and biologically characterized therapeutic agents, dose response activity with the intended route of administration, studies correlating pharmacokinetic and pharmacodynamic measures, and validation and replication studies to confirm your observed results. Next slide, please. So there are some activities that are either not in scope of the IGNITE program or fall within another IGNITE FOA. Examples of activities that are not appropriate for this funding opportunity include, but are not limited to, study or use of therapeutic agents to identify targets relevant to a disease, studies of disease mechanism, development of de novo animal models and pharmacodynamic measures, assay development and identification of novel therapeutic agents, any GLP toxicology studies or IND enabling studies with optimized therapeutic agents, discovery or development of devices, surgical procedures, diagnostics, or rehabilitation strategies, discovery and development of risk detection, diagnostic, prognostic, predictive, or prevention biomarkers, although use of existing biomarkers is appropriate. Manufacture of therapeutic agents for clinical trials, any clinical research and clinical trials are not appropriate for this FOA.
Thank you, Chardell. Next, we will have Dr. Roof talk about PAR 18762, Assay Development and Therapeutic Identification and Characterization.
So the goal here is development of new translational assays, as well as screening or identification efforts to find new novel therapeutic agents. A lot of people use this for high throughput screening, but it doesn't have to be. It can be a more focused library. It could be, biologics. It could be a lot of things. All right, now the entry criteria, the first three of these should sound familiar if you were paying attention to what Chardell had to say. It has to be novel, has to be a strong biological rationale, a strong premise. There has to be relevance for therapy development. And it also, you obviously need to have availability of test agents or a library that you will use for these efforts. You can do development and validation of the assays that you will use for your testing funnel. And we even mention here phenotypic assays. Some people are worried about phenotypic assays doing well in this mechanism, and that's not necessarily a no-go. We certainly support some phenotypic assays. There are some issues you need to think about that might be different, but certainly it's within scope. Development of in vitro or ex vivo potency, efficacy assays that you will use to evaluate your hits. Development of assays to evaluate properties. Development of assays to evaluate the purity or the identity of the therapeutic that you get out of your screening efforts. Any assay development or optimization, maybe you need to miniaturize your assay, for example, or maybe you need to tweak parameters to optimize your signal to noise or whatever. Those kinds of activities are certainly appropriate. And a combination of assays may be necessary for a robust screening effort. So we would hope that by the end of the R61, if you're successful, that your assay is developed and optimized. Perhaps you have to do physical, chemical, or biophysical characterization of test compounds that you will use for your screen. Development is selection of cell lines or vectors to produce the biologic agents, if that's what you're going to be screening. Whatever it takes in your particular project to show that this is feasible and worth going forward to the screening efforts, all right? And your milestones should reflect that. So some activities that you might do in your second phase include things like running your high throughput screen or whatever screening effort you're doing. Once you've done your screen, you might need to do some work to confirm the structure or sequence of your hit compound. You might need to assess the properties. You might need to do some computational analyses of the properties of the compound. You might want to do in vitro ADME. You might look at off-target activities, selectivity activities. You might need to optimize your agent, and by that we mean, you could do some early focused SAR efforts, whatever's appropriate for your particular project. All right, now out-of-scope activities, like Chardell said, these are mostly things that are either better supported in other programs or better supported in other IGNITE FOAs. Development of assays and probes to support basic understanding of disease, basic research. I also think of target identification or studies of disease mechanisms in this piece here. If you're doing basic research, you're better suited for a basic research FOA such as an R01, right? Recall that one of the entry criteria here was a biological premise, right? So you need to have enough understanding of the disease to justify why you're taking the approach you're taking and why you're targeting what you're targeting, or if it's a pharmacodynamic or a phenotypic screen, that you're looking at a readout that reflects something meaningful, okay? This is not a good place for basic science. All right, pharmacodynamics and in vivo efficacy studies, because that is better, that is a fit for PAR 18761, which Chardell just told us about. Okay, development of devices, surgical procedures, diagnostics, rehabilitation strategies. We have a translational devices program. If you are interested in developing devices, I encourage you to take a look at that program. Again, there's webinars, there's websites, there's people to talk to. Please use those resources. Biomarkers, again, we have a separate biomarkers program. If that's your goal, you're better served going to one of those FOAs. IND enabling studies and clinical compound manufacture, I'm going to talk about together. These are really later stage. If you're at this point, you should be talking to Blueprint or Create. If you're ready for these things, truly ready for them, you're at a later stage program. And finally, clinical research and clinical trials, with the exception of research that meets exemption 4. If you're doing clinical trials, again, you're way beyond IGNITE. Now, clinical research, this one's actually a little bit nuanced, and Jules is going to talk a little bit about this later on, so stay tuned. All right, and now I'm going to turn it over to Julia to talk about our last FOA.
All right, thanks, Becky. So the third funding opportunity in IGNITE is PAR 18763, which is for the development and validation of model systems and pharmacodynamic markers. The goals of this funding opportunity are to promote a significant improvement in the translational relevance of animal models, ex vivo systems, and pharmacodynamic markers that will be utilized to facilitate the development of neurotherapeutics. So essentially, we're looking to provide translational tools for the drug discovery process, and not just for you and your lab, but something that would really make a difference for the whole disease area, something that other investigators or companies would want to use as they're developing candidate neurotherapeutics as well. Another goal is to develop a fully validated model system, testing paradigm, or pharmacodynamic marker that can be used in both preclinical and clinical settings to test the biological effects of a candidate neurotherapeutic agent. So the emphasis here is that we want a very rigorous approach that is going to be translational, i.e. the model will mimic as much as possible the human disease or condition, and the endpoints that will be used to assess that will be endpoints that, for the most part, can also be used in a clinical setting. So overall, in addition to having a strong biological and translational rationale for your model or PD marker, there should be evidence of value for future drug discovery and development. Next slide, please. Okay, so I wanted to go over some of the relevant definitions for the context of this funding opportunity. The first is a pharmacodynamic marker is a specific type of biomarker whose levels change in response to therapeutic intervention. So this could be a component of the molecular pathway mediating the biological effects of therapeutic target modulation, which could be either direct or indirect, or it can be a component of disease etiology that is involved in drug target modulation. Internal validation addresses the consistency of the experimental design. So it involves characterizing the precision, reliability, analytical sensitivity, accuracy, and dynamic range of the endpoints utilized in the model system or biomarker measurements. Whereas external validation refers to the extent to which results of a study can be generalized from one setting to another, and these are typically thought of as three different types of validity. Face validity is the similarity between a model or model system and clinical manifestation of the disease, such as symptoms, morphology, and so on. Construct validity is the similarity between a model or model system and the physiological basis of the human disorder in terms of what is known about the disease mechanism. And finally, predictive validity is a similarity between the effect of a validated therapeutic intervention in the model and in the clinical disease population. Next slide. Okay, so some of the activities for the R61 phase include the initial development of the model ex vivo system or pharmacodynamic marker. So you can completely develop a model from scratch or take a model that hasn't been well validated and validate it. Activities also include any optimization related to the feasibility, endpoint range, or sensitivity, in all internal validation for endpoints that are going to be used, and any scale up needed for the R32 phase. In particular, for example, would be if you're developing a genetically modified animal, so you need to make sure the breeding is sufficient and you have a stable generation in order to do the studies in the R33 phase. So by the end of the R61, you want to be able to show that your model system or PD marker is feasible for the external validation studies, whether that be the actual ends for your animals or the basic internal validation studies for your endpoints. Also, you want to show that it reflects the functional or physiological pathway of the disease or therapeutic target as intended. And again, your milestones should address these aspects in a quantitative way. So examples of activities for the R33 phase, again, include all external validation studies, including comparisons of the model phenotype to human disease, comparisons of disease etiology in preclinical species to what's known about the human disease, or comparing the efficacy of clinically validated therapeutic agents in a new model system if such therapeutics are available. Next slide. Okay, activities that are out of scope for this funding opportunity include development of animal and ex vivo models for the purpose of understanding disease etiology. So again, as Becky mentioned, this would be more R01 space. Also out of scope is cell line development or the identification of CNS drug targets, the discovery and development of other types of biomarkers. Again, this funding opportunity only supports pharmacodynamic markers in particular, But as Becky mentioned earlier, I do want to point out that NINDS does have a biomarker program, and we'd be happy to put you in touch with those individuals who can further discuss the scope of their funding opportunities and potential fit for your project. So other out-of-scope activities we've already talked about, especially ones that are just different FOAs in IGNITE, and device development is out of scope, and finally, clinical research and clinical trials, which we'll go through shortly. The next slide.
Thank you, Julia. So I'm going to go through some general tips, words of advice, and these apply to all 3 FOAs. First of all, contact us in advance. We're happy to talk to you. You do not want to put all the time and effort that it takes into putting together an application only to find out that it doesn't fit with NINDS mission, or you included some out-of-scope activity and we'd have to ask you to withdraw. Like, you do not want to put all that effort in to have that happen, and we're happy to help you avoid that. So please talk to us in advance. Not only can we help you with things like FOA fit and mission relevance, we can also help you think about the kinds of things that review typically comments on or typically have issues with, and we can help you kind of navigate some of that. So please contact us in advance. You'll have our email information from the first slide here. You have Tim's email, he can connect us. Please contact us in advance. Second, have clear milestones. I hope by now I've gotten through that milestones need to be clear and quantitative and meaningful. Again, here's the link to the website that will give you example milestones. Again, this is a really common mistake that people make. So I'm trying to help you avoid that. Include rigorous study design and supporting data. NINDS has been very interested in rigor for a very long time. This FOA is absolutely no exception. Okay, reviewers take a really careful look at rigor. They will comment on the rigor. This is a very avoidable mistake. So please pay attention to rigor and make sure that that's in your application. Have a multidisciplinary team. Okay, so we haven't talked about this yet, but it's actually very important, right? So when you're doing therapy development, you don't just need the disease expertise, but you also need translational expertise, drug development expertise. You might need chemists, you might need pharmacologists, you might need a clinician to help make sure that you're going in the direction of something that's going to be meaningful, okay? Note the multidisciplinary review. So this is not a CSR basic science review panel, okay? NINDS custom makes these panels for translational research. So you will have a very multidisciplinary review. The review panel includes academics, it includes industry folks, it includes disease biologists, as well as, you know, chemistry, pharmacology, drug development, clinicians, okay? We have a very multidisciplinary review. You will likely get 5 reviewers on your application, and we do that on purpose so that we can get all of these different perspectives looking at your proposal. So your proposal needs to be written such that it satisfies all of these different multidisciplinary panel members, okay? Very important that you know your audience here. Discuss intellectual property. So this is also something that's different from your typical, you know, basic science grant. You need to discuss it. You don't need to have a patent filed at this point, but you do need to convince reviewers that you've thought about it. Convince reviewers that this isn't something that's, even if it's successful, is dead in the water because there's no way to bring it forward, okay? If you can have a letter of support from your tech transfer office, just saying that they'll work with you. Like, that's enough at this stage. Some people feel like they need to have patents. You don't need to have a patent yet. In fact, it might not even be the smart way to go at this early stage. Just show reviewers that you're thinking about it and that, you know, that this will have legs going forward. And that feeds into the therapy development plan. So this is another thing that's different here compared to some other mechanisms. You want to talk to reviewers about what happens next, okay? Where are we going from here? If this is successful, what's our next step? And, you know, where are we going? You don't need to have your manufacturer CRO lined up at this point, but you need to show reviewers that you're thinking about it. What kind of issues might you run into down the road, and what can you do now to mitigate that? Okay, are you going towards a patient population that exists, and are you fixing a problem that's important to them? Okay, these are the kinds of things you want to be thinking about. Again, you don't have to have everything lined up at this early stage, but convince reviewers that there's a path. Your multidisciplinary team will help you with this, okay? If you are a small business, I strongly, strongly encourage you to consider the SBIR program. Okay, Emily Caparello is your point of contact. Her information is here. If you are a small business, you can utilize funds that we set aside, that we're required to set aside for small businesses. And then you're only competing with other small businesses, right? That's a much better place for you to be if you are eligible. Some people feel like they need to apply to Ignite because it's very tailored to what it is they want to do, whereas in the SBIR, you would be applying to the general omnibus solicitation. That's not true. Please, if you're eligible for the SBIR program, please contact Emily and use that program. And pay attention to clinical research rules, which are not as straightforward as you might think. And I'm going to turn it over to Julia to tell you a little bit more about that.
Okay, yeah, so we wanted to briefly touch upon the NIH terms for human subjects and clinical research, since this can be a point of confusion for some of our applicants, especially when it comes to using human samples or patient-derived specimens, which has also slightly changed with the revised common rule. So human subjects research is research involving a living individual about whom data or biospecimens are obtained, used, studied, or analyzed through an interaction or intervention, or where identifiable private information is used, studied, analyzed, or generated. Whereas clinical research is defined by the NIH as research conducted with human subjects, so meets the definition of human subjects research, so it's either with human subjects or on material of human origin, such as tissues and specimens, unless those studies specifically fall under the CFR's criteria for exemption 4. So federal regulations include 8 different types of exemption, and clinical research includes everything except for exemption 4. So what is exemption 4? It involves the collection or study of data or specimens if the sources are publicly available or the information is recorded such that subjects cannot be identified. But the key is that exemption 4 is for secondary research only. If the biospecimens are being collected specifically for the research proposed in the grant through an interaction with living individuals, then it's considered primary research. So overall, depending on the specifics of how your samples are collected and how they're coded or de-identified, will determine whether it qualifies as human subjects research at all, and whether, if it does, does it meet the exemption 4 criteria. Now, NIH's Office of Extramural Research provides a number of useful resources through their website, which we provided here and would encourage you to visit. Importantly, if you're unsure about the designation of your research, we highly recommend reaching out to your institute's IRB or human subjects officer, because they're going to be the ones who are ultimately making this designation, not us. You can also email OER at the address provided here for guidance. And if you go to the next slide. So this is just the homepage for the website that I was referring to in the last slide. In particular, I just wanted to point out their decision tool, which guides you through a series of questions, and we found this to be quite helpful, so we definitely encourage you to use it. Okay, next slide. Okay, so we're going to go through the questions. I'm going to read the questions, and then Becky is going to answer them, although I might jump in every once in a while. But you're welcome to continue asking questions in the Q&A session, and if we run out of time, we'll still try to address them as we can. So, all right, Becky, you ready?
Yep, let's go.
Okay, so the first 2 questions are about funding. Can any of the funding be spent at contracted research labs, and can any of the funding be applied to salary?
Yeah, absolutely. Salary for sure. Things to CROs, that's allowed. I would be careful if you're sending so much to the CRO that it impairs your ability to do other things, because there is a limit on how much budget you can spend. but those things are certainly allowed.
Which diseases or disease areas are being prioritized?
So for IGNITE, anything that is within NINDS mission is fair game. NINDS has in the past been given special funding for things like pain or ADRD, but you don't necessarily get any change in our IGNITE portfolio for those things. Those are typically done through special RFAs, although there can be exceptions. So I would say anything within NINDS mission.
Okay. And how many awards are expected to be funded?
So we typically fund between 4 and 6 in any given council round. But an important point, and we probably should have said this before, is these are not pay line driven. You won't get a percentile on your grant. We really pay a lot of attention to what's in the summary statement, as well as things like, likelihood of success and fit for the FOA. So if we, for some reason, got a really high number of really awesome projects, we would be in a position to fund more. If for some reason we had a really low number of really great projects in a given round, we would fund less.
Okay, what level of preliminary data are expected in these applications?
So it's kind of hard to answer that in a blanket statement, but I'll do my best. So if you look at the entry criteria for the different FOAs, premise for the disease, premise for your approach, you do need to convince reviewers that you're targeting something that's meaningful, right? So whatever that takes, they will also be interested in some idea that what you're doing is feasible. That's the best I can do on a blanket statement, but if you want to contact me with your particular project, we can talk about it further.
Okay, is this for new drugs only, or can this be applied for repurposing of existing drugs?
Oh, absolutely, you can do repurposing. We see quite a bit of that.
Okay, so a couple SBIR related questions. What criteria a small business should consider while applying for this grant, and if there is a direct comparison between the IGNITE program to the SBIR STT program.
Okay, well, so if you're a small business, again, I would strongly recommend you look at the SBIR program instead of IGNITE for the reasons I talked about. If you do choose to go to IGNITE, your entry criteria are the same as anybody else. I know the SBIR program has some pretty nuanced criteria about how much work can be done internally versus externally and all of that. None of that applies here. With the SBIR program, you will need to have a commercialization plan. Here you will not, although you need to have a therapy development plan. So you still need to be thinking ahead. But again, if you're an SBIR eligible company, you're way better off applying to the SBIR program because of the set aside funds.
Okay, do IGNITE applications go to a particular study section or just that which is applicable to the focus of the proposal?
All right, so I think we answered this already, but I'll say again, we have an internal NINDS review for these with all of the special expertise that I talked about before. They do not go to CSR.
Okay. So the next question is both vague and specific. It says we have been working with a mutated growth factor that has great translational potential. What will be the FOA that will fit? And this is very specific to your project, and it depends what you want to do with it. So this is something where we would recommend you contact us, send us your specific aims page, and set up a call and we can discuss.
Thank you.
Next question is, can we work with foreign collaborators?
You can. You will need to justify it. Just like any other mechanism, you need to justify why you need to have the foreign collaborator, and it can't be done domestically. Now, if a foreign collaborator is easier than a foreign grant, if you're proposing to do a foreign application to a foreign institution, the bar is actually quite high for justifying why it needs to be done at a foreign site and can't be done domestically. you're allowed to apply, but just know that the bar's higher for a foreign grant. Now, foreign component, as long as the justification is reasonable, that's okay.
Can we propose to do the chemistry optimization ourselves?
Absolutely.
Okay. Can we partner with NCATS on our project? We are already collaborators.
Hey, that's great news. Yes, you can absolutely partner with NCATS. It's a great resource. The only caveat is we cannot pay them, so you can't request, salary or any other support for NCATS on your grant. But absolutely work with them. They're a great resource.
Next question is asking, how do we initiate interaction with IGNITE? And that's pretty simple. We've given you our contact information. You can email us and we can set up a call if you want to discuss the particular project.
Yep.
All right.
Please send us an AIMS page in advance. We're much more help to you that way.
Yeah. How close would we have to be to clinical use? Validating a biomarker generally takes hundreds to thousands of patients. I agree. So again, we have a biomarker program that's actually geared towards that later clinical stage, but everything at IGNITE should be preclinical. And the restriction on the use of human subjects unless it qualifies for E4 also sort of keeps you in that mostly preclinical stage. Does IGNITE also apply to biologicals that go through CEDAR?
Yep. Yeah, we support biologics as well as small molecules.
Okay. Could you please elaborate the main difference between 761 and 763?
So 763 is for animal model development. 761, we can even go back here. Let's go back to that slide here. Oops, I went too far, I apologize. Oh, no, I didn't. Anyway, here we go. 761 in vivo efficacy studies. So if you're testing a therapeutic, a potential therapeutic, you would use the in vivo efficacy studies. If you're developing your animal model, you would use the 763.
So then a follow-up to that would be, would 761 include PET ligand development to enable receptor occupancy preclinically or clinically?
No, actually. So we're going to go to the on-scope activities. Development of biomarkers, the third bullet from the bottom, is not in scope. If you have existing biomarkers, you may use them.
Yeah, so you could potentially develop the PET ligand in 763, but that will depend on the specifics. So again, we would recommend you reach out to us.
Absolutely.
Okay.
The budget might be an issue for you there too, but we can talk about it.
Yeah. Would drug device combinations be able to apply to IGNITE?
Unfortunately, no. Those would be better suited in the device mechanism.
Okay. Do the activities of conducting a pilot screen for a target with new developed pathway assays and looking for the hits in vivo physiology outcomes suite, or I guess fit in PAR 18762? Sorry, that was worded a little awkwardly.
Yeah, so pilot screening certainly you can do in 762. Some people propose to do that in the first phase, and that's quite appropriate. What was the other part of that, Jules?
I think it's looking at the hits in vivo physiology outcomes. Is that also in scope for 762?
Okay, so in vivo efficacy of your hits is actually out of scope. That's a better fit for 761, where you could spend the full 3 years doing a proper proof of concept with the PK and whatever else you need to do to make sense of it.
What is the deadline for resubmission of a proposal?
That's a great question.
Been previously reviewed. Go ahead.
Yeah, I'm glad that you asked that because it seems like every round we have somebody who has a problem here. The A1 dates are the same as the A0 dates, okay? We don't have that month delay in this mechanism that you might see in like an R01. Okay, so I'm glad you asked that because you need to know it's the same date, you don't get a delay.
Okay, regarding later stage programs, even at this stage, there is a lot of PK/PD and SAR research taking place. Mm-hmm. Why can't a program be eligible even if it approaches an IND?
I guess I would have to understand a little bit more about the project and why they were proposing to do IND. Maybe that's somebody who we should talk to individually about.
Yeah, because some of these PK/PD SAR studies can also be done in BPN depending on the track you're at.
Yeah, absolutely. Yeah, if you already have your proof of concept, right, that gets you the entry criteria for Blueprint or Create, and you're right, that might be a better suited. But again, it depends on your specifics. So please send us an email and we'll talk about it.
Are compound mechanism of action considered part of IGNITE, or does this fall under basic biology?
Yeah, so the devil's a little bit in the details there. So is this something that is absolutely critical for moving this forward, or is this a curiosity exploration activity, right? So especially if you're doing like a phenotypic screen in 762, the assay development screening FOA, you might need to do some mechanism of action understanding to understand what your target is in order to bring it forward. And in that case, it would be fine. If you are exploring mechanisms because you're interested in how that interplays with the disease and what that means about disease biology, that's not going to fit. So it kind of depends.
Okay, we have a couple questions related to SBIRs in terms of their phases, distribution of work effort, how it compares to an SBIR, STTR application. So I'm just going to sort of try to answer all of these together in that. I mean, again, we can't reiterate this enough. We would highly encourage you to use the SBIR funding. So small businesses are eligible to apply for IGNITE, but you're not using SBIR funding, so you're not following any of those SBIR rules. So it's going to be the more standard application instructions that are included in the funding opportunity. Okay. One question, if you're also submitting SBIRs, does that exclude you from applying to any of these 3 FOAs? So it depends on the timing, and this is not for SBIRs in particular, but for anyone. If you're applying to a funding opportunity at NIH, you cannot submit another application with any significant overlap in the aims until you receive the summary statement from the first application that's already in the system. So you can't have overlapping things under review at the same time.
Unless one of them's an RFA, which probably isn't the case with an SBIR and certainly isn't the case here.
Yeah. Okay. Regarding 18763, can you comment on the threshold for understanding the disease etiology? How well does the disease etiology need to be understood?
Well, that's kind of a review question, and it's going to depend on, the disease and the model, and you need to convince reviewers that you know enough to make a model that replicates disease, because that's a requirement. So I guess it kind of depends on the details.
Are non-U.S. citizens and non-permanent residents qualified to apply as a primary PD or co-PI?
I'm actually not 100% sure on that one. I would have to look it up and get back to you.
Yeah, so the funding opportunity will list the eligibility criteria, and those for IGNITE are pretty standard in terms of what would be for R01s and such. So, because it's actually the institution that's putting in the application. So there are eligibility requirements in terms of what constitutes a PI.
Yes, thank you, Jules.
Okay. Can IGNITE be used on programs of interest to NIA, for example, Alzheimer's or cognitive decline?
NIA does not participate in IGNITE, so anything that you put in would have to be with an NINDS mission.
So we do recognize that with the ADRDs, there is a bit of overlap. So this is something where we would prefer you contact us before you try to submit to make sure that what you want to do is within mission.
Yep.
Okay.
We have 1 minute left.
Oh boy.
Yeah,
Let's see.
Can you merge a few?
I'm trying to pick out, but fine question. Would this be appropriate for rare diseases? Yes. Anything
Absolutely.
within NINDS's mission. So pediatric brain tumors, yes. It's something we haven't addressed yet. To what extent would a multidisciplinary team be required for development and optimization of a novel high-throughput screening strategy that targets a previously defined protein target and which is validated by published in vivo studies in a knockout mouse? This one sounds like a very specific question, so I think
it does.
Multidisciplinary
Multidisciplinary teams are always a good idea, though. We'll just leave it at that.
Will the IGNITE program be renewed? For sure, as the current expiration date is May 2021.
Oh, somebody's looking ahead. That's great news. I can't comment on that.
Can you pay for an investigator to be consultants?
Yes. You can have consultants.
Assay development and therapeutic identification often involve characterization of a class of compounds. In this regard, there are some overlaps between 761 and 762. Can you clarify, and will overlaps disqualify the application for review?
Okay, so that's actually a good point. So we allow some characterization, optimization, SAR at the end of a screening effort. We also allow a little bit of optimization and characterization at the beginning of 761 before you do your efficacy studies. And that's actually on purpose. So that gives you a little bit of wiggle room about where you're at before so you can, that's on purpose to give you a little bit more flexibility. And that's fine.
Yeah. Yeah, the difference becomes in 762 when you want to start doing stuff for efficacy in particular. That is out of scope. So, okay, I think that's all the questions that we have. We have some more, but they're all repeat questions. So
great. Fabulous. Hey, Tim, do we need to do anything to close out here?
No, I just wanted to thank everyone for joining. And like I mentioned earlier, we will be sending out everyone a copy of the slides. I'll probably do that by tomorrow morning. And then the hope is that we get the recording to everyone, at least everyone that's registered to this webinar by next week, and then post to our website within a month. There's a little bit of compliance issues that we have to go through in order to post it to our website, but they're not as stringent with us emailing it. So I'll make sure to get the recording to everyone, hopefully by the end of next week. If you have any suggestions on ways we can make this webinar more efficient, or if you have, or better, or if you have any other thoughts on any subjects you'd like us to do, please let me know. My email address is on your screen right now. Other than that, thank you very much. And once again, we really do appreciate you joining us, and we hope everyone is safe.
Thank you, Tim.
Right.
Appreciate your help with this webinar.
Bye-bye.
Bye-bye.